Good morning and welcome to this lesson on DNA profiling. So today we're going to be looking at this technique and really we're going to try and understand the principles behind DNA profiling and also some uses of this technology. So to start with, I'd just like you to answer this question. Who did it? Explain how you know. Got some image in the left corner here. Who did this crime? Okay, I imagine that you worked it out pretty quickly, but how did you do so? Well, it's suspect 2. And the reason that you knew that was because you matched the pattern of the crime scene to the pattern of the suspect. So really DNA profiling involves matching this banding pattern. Now you can do that without knowing much about the technique at all, but we're going to be looking at how this banding pattern is produced and why this banding pattern is different in different people and how we can be sort of sure about that. So the person who came up with this technique was a guy called Sir Alex Jeffreys, now Professor Sir Alex Jeffreys. He came up with it in the 1970s, and in a minute we're going to go into detail, but first of all, just as a sort of overview, what he did was he took DNA samples, cut them with certain enzymes called restriction enzymes, which you should be familiar with from GCSE, and chopped up the DNA into different length fragments. Then he separated them by gel electrophoresis. We've met this a little bit already when we were looking at our topic of DNA sequencing the other day. I'm going to go into more detail next lesson. So he used gel electrophoresis to separate those fragments. And then he used a special form of staining to produce this pattern. So now is your time to try and get into this technique a little bit more detail, and I'm not going to reinvent the wheel here because there's 2 excellent videos about this process. First of all, there's this DNA fingerprinting Bozeman science video, which you should watch. That's probably my preferred one, or you can watch this Mr. Exum video. They're both about the same amount of time, so I'll put the links underneath this video. Go ahead and watch one of those videos. You don't have to take too many notes because I've got an activity coming later which will test your understanding of that. Just try and get a feel for the technique. So once you've watched that video, just one of them is fine. Come back to this and we'll continue the lesson. Okay, so pause this, open it in a new window, off you go. Okay, so did you understand the video? You can always watch it again, or you can pause it or go back if you need to. But what I'd like you to do to sort of test if you've got it is to do 2 things. First of all, I'd like you to just copy this diagram just to kind of have a image that represents the technique in your notes. It doesn't have to be super artistic, but just give it a go. And then the second thing is I'd like you to take these statements here and arrange them in the correct order. So they're not in the correct order now. See if you can write them out in the right order to describe how this technique works. Pause the video, take you a couple minutes, and then come back and I'll give you the answers. Okay, here we go. So this should be what you wrote down. First we get the DNA sample, and then we have to copy the sample. We have to copy it up using a method called PCR, or polymerase chain reaction, which we're actually going to look at in our next video next week. Then we cut the DNA using these restriction enzymes. They're kind of like molecular scissors at specific sites. And the fragments are cut to provide DNA of different lengths from different individuals. The fragments are separated using gel electrophoresis, and then we see that the shorter fragments travel faster and further in the gel, and the longer fragments travel more slowly and not as far in the gel. Then we can look at this banding pattern of the fragments by using special stains, and then we compare it with other people. So that's this DNA profiling technique. But one thing which you might be wondering is, how come people have these fragments of different lengths? How can we be so sure? Well, we're going to look at that in a second. First of all, sorry, I forgot this question. First of all, some follow-up questions on that activity. You may need to look over page 221 and 222 for help, but see if you can answer these 3 questions below. Pause the video and have a go. Okay, did you get it? So, the answers to these questions. First of all, what type of DNA is used for this analysis today? Well, it's principally non-coding DNA. Okay, and that kind of leads into question 2. Why would you not look at a coding gene when doing this analysis? And that's because coding genes would show similarity between all people. I mean, we all have let's say, for example, the gene hemoglobin. The vast majority of people have exactly the same genetic code for hemoglobin, which makes the functional protein. Some people might have one or two differences, but that would most likely cause disease. For example, sickle cell disease would have a slightly different version of that hemoglobin gene. So 3, what are the odds of getting a random match between two individuals? Well, it's really, really, really small. It's about 1 in 10 to the minus 1 in 10 to the 18. So that is just like, I can't even put that into numbers. I think that is a billion. Yeah, so 1 in a billion. So that is vanishingly small. Okay, so what about the history of these different techniques, the history of the DNA profiling and the different types of DNA sequence that we've used to do it? So there's a lot of kind of letters that we use, like RFLP, AFLP, and STR. And all of these acronyms are just words for different forms of repeating segment in the DNA. So in the 1970s, we used something called restriction fragment length polymorphism, but that's quite a lengthy process. In the 1990s, we started using something called amplified fragment length polymorphism, and in some countries that's still the main technology used. Today we use short tandem repeats. And we actually look at 20 different places, 20 different loci in the chromosome, across the different chromosomes that are inherited independently. So that means that they're on different chromosomes, so they don't show linkage. And so there's the probability of 1 times 10 to the minus 18 of a match for two people that are not identical twins. So it's pretty accurate. That is a really, really small probability. So the readout that you get actually today would look something a bit like this. We don't actually use gels anymore. We use computer software to kind of analyze those fragments. And here's an example of a matchup. So above we have the mixture from the evidence, and below we have the suspect. And we can see that the peaks match up exactly. I can see there are some peaks here from the evidence sample that don't seem to be this suspect. For example, here doesn't seem to be anyone there. Maybe that's someone else at the crime scene, or maybe that is, if there was a victim in this crime, maybe that's the peaks from the victim, I'm not sure. So what are the uses of DNA profiling? Okay, I'm going to draw a little mind map and I'd like you to copy this down. Let's have a look. So there's many uses, basically. Here's one. It's been used to identify war criminals hiding in South America. So after World War II, quite a lot of Nazi generals tried to flee to South America, and after many years they were sort of found, but it was difficult to prove that they were who we thought they were. So this has been used in this instance. It's also been used to identify Richard III, who was found in the car park, I think in Leicester, I think it was. Let me just check. I think he was in Leicester or Leeds. Anyway, his remains were dug up under this car park, and looking at the DNA from the skeleton and comparing it with DNA from known sort of descendants of his, we actually found out that this was Richard III that was found in a car park in Leicester. Then we've got also this girl here. This girl was claiming to be someone called Anastasia, who was a descendant of the Romanov family. So the Romanov family were the Tsars of Russia, and during the Russian Revolution, they were mainly killed. But this girl was claiming to be a descendant, the lost sort of princess Anastasia. But actually, after she had passed away, after she died, it was found that she wasn't who she was claiming to be, because her DNA profile didn't match that of her the close family samples that we had. It can be also used to match relatives to unknown grave sites. For example, in this military cemetery, I think that might be in France. And we can also use it to identify crash survivors as well in plane wreckages if there are no other records. Other ways, of course, are using things like dental records and things like that. But if the remains are quite degraded, then that's the sort of thing that can be done. Okay, so there's another use of this technique, which I'd like you to sort of figure out here. Have a look at this image. What do you think's going on? Pause the video for a second and see if you can work it out. All right, did you figure it out? Well, this is actually a paternity analysis. Okay, so we're looking again at these short tandem repeat sequences in a child and comparing those to the mother and then to males. So in excuse me, in a DNA profile, these banding patterns either come from the mother or from the father. 50% from the mother, 50% from the father. So therefore, we can identify maternity and paternity based on this analysis. So have you figured it out? Have you figured out which man is the father of this child? Hopefully you have. It is male number 1. So what you can see here is the blue bands in the child's DNA profile come from male 1, and these pink bands come from the mother. Remember, 50% of the STR sequences come from the mother and 50% from the father. Here's another one for you to work out. Pause the video and identify who's the father in this paternity analysis. Is it F1 or F2? Okay, here it is. So I've just highlighted that. The yellow ones are from the mother and the blue bands are from the father. So there's one other use as well of this, and it's analysis of disease. So sometimes we can do this analysis on coding regions. So one example is when we look at the gene for the protein Huntington. Now you might remember this from our lesson on genetics. So this gene Huntington contains a repeat, CAG, CAG, CAG. So it's a triplet nucleotide repeat, and it's usually present fewer than 35 times in this protein Huntington, just back to back. But if it's repeated more than this, it can lead to Huntington's disease. So here's a gel electrophoresis showing this repeat, basically, and we can see here's a scale, so 48 repeats, 56 repeats, 88 repeats. So all these people actually, I think, would have the Huntington's disease, but people with many repeats, like 56 or 88, would generally have a more severe version of this disease that would start earlier as well. So this shows the level of repeats and sort of the disease status. And just to go a little bit deeper about why that is, well, we're not fully sure, but here's the protein Huntington, the normal protein Huntington. But if this repeat segment is longer, then it kind of produces, I don't know where exactly on the protein, but let's say right here, for example, it produces a kind of big kind of coil that loops out of the protein, and that leads to aggregation, so different proteins clumping together and clustering like this. Here's a picture of a protein aggregate, so Huntington protein sort of aggregating together, and this basically messes up the neurons in the brain and leads to atrophy. So we have a lot of sort of dark space in this MRI of the brain, and especially in here, which basically means that brain cells have died, and this produces the symptoms of Huntington's disease. Okay, so that's pretty much it. That's DNA profiling finished. So what I'd like you to do now is to pause the video, work through these 3 questions, and then come back when you've answered them, and we'll see how you did. All right, here are the answers. So green pen that work, please, and send a photo of it to me on Teams. But before you go, let's just quickly do a syllabus check. So here's the syllabus. Do you understand the principles of DNA profiling and its uses, to include forensics and analysis of disease risk? So that's that last bit. Disease risk was the Huntington's bit we just did. And I just left these other 2 bullet points here as well, because we're kind of going to go through in the next couple lessons. So polymerase chain reaction we're going to do next lesson. And then we've also started to kind of understand uses of electrophoresis for separating DNA. We did that today. And also for separating proteins, that's the Huntington thing. And we've kind of also looked at electrophoresis in terms of sequencing as well, but we're also going to do another lesson on that. Okay, so that's the syllabus check for today. I hope you've found that video useful, and I'll post another video on Monday. Thanks.